The gut may be tucked away in the abdominal cavity, but its influence travels far, reaching your skin, your joints, your immune system, your brain, and especially your bloodstream. Welcome to the gut-blood axis, a molecular highway where trillions of microbes in your intestines manufacture tiny compounds that pass into circulation, subtly or dramatically altering your health. Far from being just a digestive oddity, this axis is emerging as one of the most powerful levers of systemic aging and disease prevention. Whether you're thriving or declining, chances are your bloodstream reflects the chemical chatter of your microbes. Let's start with the basics. When you eat, you're not just feeding yourself, you're feeding your gut microbiota. And these microbes are master chemists capable of fermenting fiber, metabolizing polyphenol, and breaking down amino acids into an astonishing array of bioactive molecules. Some of these are absorbed into the bloodstream, where they can reduce inflammation, improve insulin sensitivity, regulate immune function, and even influence gene expression. One of the most studied classes of these compounds are the short-chain fatty acids, SCFAs, primarily butyrate, propionate, and acetate. Produced when gut bacteria ferment dietary fiber, these molecules aren't just a local energy source for colon cells. SCFAs enter the blood and act like molecular messengers. Butyrate, in particular, has epigenetic effects. It inhibits histone deacetylases, DACs, changing the expression of genes involved in inflammation, aging, and cancer resistance. It also helps preserve gut barrier integrity, which in turn reduces the leakage of harmful substances into the bloodstream, a key trigger of systemic inflammation. In animal models, butyrate has even been shown to extend lifespan and protect against cognitive decline. Not bad for something your gut bacteria made from leftover plant fiber. Then there are the indoles, derived from the microbial metabolism of tryptophan. Compounds like indole 3, propionic acid, IPA, and indole 3, aldehyde cross into the bloodstream and activate host receptors like the aryl hydrocarbon receptor, AHR, and pregnane X receptor, PXR. These interactions influence oxidative stress responses, epithelial integrity, immune modulation, and even neuroinflammation. IPA, for example, is a potent antioxidant and neuroprotectant. Low levels are associated with accelerated cognitive aging and neurodegeneration. On the flip side, not all microbial byproducts are helpful. Under conditions of dysbiosis, an imbalanced microbiome often caused by poor diet, antibiotics, or chronic stress, gut bacteria can produce pro-inflammatory and toxic compounds that flood the bloodstream. One notorious example is lipopolysaccharide, LPS, a component of the outer membrane of gram-negative bacteria. If the intestinal barrier becomes compromised, a phenomenon often referred to as leaky gut, LPS can seep into the blood and trigger systemic inflammation. This is the essence of metabolic endotoxemia, a condition now linked to insulin resistance, obesity, cardiovascular disease, and accelerated biological aging. Other microbial metabolites, like p-cresol, hydrogen sulfide, and certain secondary bile acids, can also become problematic at high levels. P-cresol, for example, is a tyrosine metabolite that, when elevated, contributes to kidney dysfunction and neurotoxicity. Meanwhile, excessive production of harmful bile acids by gut bacteria may damage intestinal cells and disrupt cholesterol metabolism, raising the risk for liver disease and colorectal cancer. Interestingly, the host's genetics and lifestyle, including exercise, fasting, polyphenol intake, and sleep quality, can all shift microbial behavior and thereby change the blood's chemical profile. For instance, diets rich in fermented foods or polyphenol-heavy fruits, like berries or pomegranates, encourage bacteria that produce beneficial uremic toxin-reducing enzymes, SCFAs, and anti-inflammatory indoles. This microbial shift can be seen in blood tests as reduced inflammatory cytokines, improved insulin sensitivity, and elevated markers of mitochondrial activity. The implications of this are vast. The gut-blood axis doesn't just passively reflect your health, it actively drives it. It means that your microbiome isn't simply digesting food. It's writing molecular letters to your brain, liver, skin, and immune system, letters delivered through your blood. And you, through your diet, stress levels, and environment, are co-authoring the message. In terms of anti-aging, this axis offers a compelling new paradigm, Longevity may not come solely from targeting human genes or cells, but from supporting the microbial ecosystem that supplies your bloodstream with protective molecules. Therapeutic interventions could range from precision prebiotics designed to stimulate butyrate or indole production, to postbiotic supplements that bypass the gut and deliver microbial metabolites directly into the blood. There's even speculation that future, youthful blood therapies might involve mimicking the molecular signature of a healthy gut, minus the transfusion. So, the next time you think of your gut, don't stop at digestion. Think of it as a bioreactor, quietly influencing the chemical tone of your bloodstream every moment. Whether it's helping your cells regenerate, stoking inflammation, or modulating your metabolic destiny, the gut-blood axis is a powerful mediator of your body's aging process. And it's one you can influence meal by meal, microbe by microbe.